Good afternoon to all the NEET aspirants 2026. This is your Prasanna Ma'am from your NEET World. So, today we are going to exercise the rest questions from electron transport system of the chapter photosynthesis in higher plants. So, yes, let us get started without any delay. So, first question, if you see, bacterial photosynthesis contains what? Bacterial photosynthesis, which is also called as anoxygenic photosynthesis beta, okay, anoxygenic photosynthesis, but because there is no oxygen evolved from bacterial photosynthesis. And there is one more important question, that what is the electron donor of bacterial photosynthesis? Electron donor will be H2S, okay. So, H2S will be the electron donor. So, oxygen is not evolved from photosynthesis. When oxygen is not evolved from photosynthesis, what do you call that type as? Bacterial photosynthesis or anoxygenic photosynthesis. In this, only photosystem 1 is involved, that too over stroma lamellae, over stroma lamellae, only photosystem 1 will be present, okay. What is present, ma? Photosystem 1 will be present above the stroma lamellae, and this photosystem 1 will be conducting cyclic photophosphorylation, wherein the sunlight in the form of photons are accepted by ferredoxin, which is submitting to cytochrome B6 F complex and then to plastocyanin. In this way, it will be synthesizing ATP. Okay, sorry, here ATP is being synthesized, and from plastocyanin back to ferredoxin. This will be the cyclic photophosphorylation that is happening in bacterial photosynthesis. So, with respect to bacterial photosynthesis, what you have to remember, beta? Bacterial photosynthesis, also called as anoxygenic photosynthesis, where H2S will be the electron donor and photosystem 1 is present in the stroma lamellae, which is conducting cyclic photophosphorylation. So, here the answer will be what? Here the answer will be bacterial photosynthesis contains only PS1, photosystem 1. Next, during non-cyclic photophosphorylation, electrons are continuously lost from the reaction center of photosystem 2. Which source is used to replace these electrons? So, to answer this question, you should have this basic idea that we have discussed that this will be the thylakoid lamellae or thylakoid membrane, okay, thylakoid membrane or thylakoid lamellae, and this will be what? Thylakoid lumen. In the thylakoid membrane, you will be having both the photosystems PS1 and PS2. Sorry, PS2 is first and then PS1. How this non-cyclic photophosphorylation is activated? We know in this photosystems what is present, ma? Light harvesting complexes are present in the photosystems. So, this light harvesting complexes will be having accessory pigments or antenna pigments and the core pigment as well. So, here the accessory pigments or antenna pigments will capture the light energy at different wavelengths, and that will be submitting to what? Core pigment. So, here when the electrons are getting excited and submission is happening to the reaction center, that is chlorophyll A, the continuous supply of electrons is mediated by the photolysis of water inside the thylakoid lumen, where water molecule is being photolysed to 2 protons plus 2 electrons and an oxygen atom. Clear, ma? So, from here electrons are being continuously supplied to the reaction center. That is how non-cyclic photophosphorylation is activated. Non-cyclic photophosphorylation occurs where? In the thylakoid membrane. So, if this I have to draw over here, if you can see, in this thylakoid membrane, light in the form of photons will be activated, will be received. and then these are submitted to the reaction center, that is chlorophyll A. From the reaction center, 2 electrons are accepted by primary electron acceptor. That primary electron acceptor is plastoquinone. Okay. This primary electron acceptor is pheophytin, primary electron acceptor is pheophytin. Pheophytin will take 2 electrons and it will be giving to plastoquinone. Okay, plastoquinone will be taking 2 electrons, accepting 2 electrons, it will accept protons also, 2 protons also from the stroma. Then it will be converted to PQH2, plastohydroquinone. This H plus is being sent to the thylakoid lumen and electrons, 2 electrons are being given to cytochrome B6F complex. From here, the last electron acceptor of photosystem 2. This is what photosystem? This is photosystem 2 from here to here. So, the last electron acceptor will be plastocyanin. From here, the electrons are being again moving in the uphill motion and the reaction center here has maximum wavelength of P700. Okay, maximum wavelength is P700 and here the primary electron acceptor will be ferredoxin, ferrous sulfur complex, and lastly over the thylakoid lamellae NADP plus is present on the outer side of the thylakoid membrane. This is the ultimate electron acceptor of non-cyclic photophosphorylation. This NADP plus accepting 2 electrons, this gets reduced to NADPH plus H plus. Okay. This is a gist of non-cyclic photophosphorylation. Clear ma? So, this is what photosystem 1 here. Photosystem 1 and photosystem 2 both are present in the thylakoid membrane. Okay. Both are present in the thylakoid membrane of oxygenic photosynthesis. In those plants where oxygenic photosynthesis is happening, photosystem 2 and 1 are present over the thylakoid membrane, and this flow of the electron transport you have to remember. First is uphill, then downhill, uphill and then downhill. Uphill is when electrons are getting excited to the reaction center, downhill is when they are carried through the protein carriers. Okay. First primary acceptor is pheophytin, then plastoquinone, cytochrome B6F, last electron acceptor of photosystem 1 will be plastocyanin. Then again from photosystem 1, ferredoxin, ferrous sulfur complex, the ultimate electron acceptor of non-cyclic photophosphorylation will be NADP plus. Clear? So, this gets converted to NADPH. Now, it will be easy to answer this question. Okay. This set of questions, look into it carefully. First, during non-cyclic photophosphorylation, electrons are continuously lost from the reaction center, that is PS2. What is the source used to replace this lost electrons? Here you can see, no, to replace the lost electrons, what is the source? Water. Okay, because this gets photolysed and this is continuously supplying the electrons to the reaction center of photosystem 2. So, here what will be the answer beta? Water molecules. Clear? So, this is the source used to replace the electrons. Next, here you see, read the following statements and select the correct ones. Okay, read the following and select the correct ones. These are statement based. So, here if you see ma, PS1 photosystem 1 is involved in non-cyclic photophosphorylation only. Wrong. Just now we have discussed, right? PS1 which is present in the stroma lamellae is conducting only cyclic photophosphorylation, that too in bacterial photosynthesis. PS2 is involved both in cyclic and non-cyclic. No, PS2 is involved only in non-cyclic, not in cyclic. So, this is also wrong. Third statement you see beta, stroma lamellae membranes possess PS1 only, whereas grana lamellae possess both PS1 and PS2. Correct, right? grana lamellae or thylakoid lamellae, one and the same. These membranes are having two photosystems, photosystem 2 and photosystem 1, whereas stroma lamellae has only photosystem 1, that is conducting bacterial photosynthesis. So, now here he asked us to identify the correct ones, and the correct statement is option 3. Clear? Next question. In the above schematic diagram, which is plastocyanin? Okay, so he is asking us where is plastocyanin present. Just now we have clearly discussed about the electron flow, right? So, from PS2 reaction center, the primary electron acceptor will be pheophytin, next it will be plastoquinone, and then it will be cytochrome B6F, and then it will be plastocyanin. From plastocyanin, the electrons will be accepted by PS1 reaction center in uphill way. Here the reaction center is P700. Clear, ma? So, now here you tell me in this diagram which is plastocyanin? D will be the plastocyanin, option D. Clear? Next, steps in non-cyclic photophosphorylation. Okay, what are the steps involved in non-cyclic photophosphorylation? Include the passage of the electrons, how the electrons are being passed. Of course, first if you consider FRS to ferredoxin, no, because these are involved in photosystem 1, but the electron flow starts from photosystem 2. So, that will be wrong. Next, if you have to consider this one, option B, chlorophyll A to cytochrome B6F, then cytochrome F he is saying, wrong. From chlorophyll A reaction center, the primary electron acceptor will be plastoquinone. So, we cannot choose this and we cannot choose this as well. This you see plastoquinone to cytochrome B6, then cytochrome F, then plastocyanin, and from plastocyanin to PS1 FRS ferredoxin. Clear, right? So, there is a clear flow from photosystem 2 to photosystem 1. After ferredoxin, it will be ferrous sulfur complex, and then the ultimate electron acceptor will be NADP plus. So, here what will be the answer beta? Option D will be the answer. Clear, right? Next question you look into assertion and reason. Cyclic photophosphorylation synthesizes ATP. Clear, right? Cyclic photophosphorylation is synthesizing ATP, that too in stroma lamellae. Okay, it is synthesizing in stroma lamellae. Fine. So, cyclic photophosphorylation synthesizes ATP where ma? Inside the stroma lamellae. Assertion is true. Reason: ATP is synthesized in cyclic photophosphorylation is not associated with NADPH formation. True. Okay. Where in the cyclic photophosphorylation you can see only ATP synthesis but not NADPH formation. So, reason is true, but in reason it is not explaining why in the cyclic photophosphorylation only ATP is being synthesized. Okay. So, clearly once again you read ma assertion: cyclic PP synthesizes ATP, correct. ATP synthesis in cyclic photophosphorylation is not associated with NADPH. So, both the statements are true, but in reason it is not explaining why only ATP is being synthesized. In that case, what option we have to opt for? Both assertion and reason are true, but reason is not the correct explanation of assertion. Next, what does the given diagram represent with respect to the various photosynthetic processes? So, clearly looking into here where light is accepted by P700, that to photosystem 1, and it is cyclic, only ATP is being synthesized. Looking into that only you can say it is the cyclic photophosphorylation, right. So, here what does the given diagram represent? Of course, it is cyclic photophosphorylation, because there is no involvement of P680, there is no involvement of photosystem 2. In that case, definitely it will be cyclic photophosphorylation only. Next, you look into ma recognize the figure and find out the correct matching. We have to identify the correct matching. Here you see first A will be of what? First from light harvesting complex, as two light harvesting complexes are there. This is a definitely non-cyclic photophosphorylation. From light harvesting complex, first electron acceptor, that is photosystem 2. Fine, this is what? PS2, A will be PS2. And here if you see, ADP and inorganic phosphate will form ATP. So, D will be ATP. Next B, if you see, B is what ma? Photosystem 2. This is B is photosystem 1, because here it is P700, second light harvesting complex P700. And here from NAD plus, that gets reduced to what? Ultimate electron acceptor from Z scheme. This gets reduced to NADPH. Fine, NADPH. So, here if you identify the options clearly, A is what? Photosystem 2, B will be photosystem 1, C is NADPH, D will be ATP. In that case, the right option is option D, where A is photosystem 2, B is photosystem 1, C is NADPH and D will be ATP. Clear ma? Right. So, this is how you have to recognize the figure and identify the option. During non-cyclic photophosphorylation, electrons are continuously lost from the reaction center, that is photosystem 2. Okay. From photosystem 2, electrons are continuously lost from the reaction center. Which source is used to replace these electrons? Okay. To replace these electrons, what source is used? Of course, it is water. Just now we have solved. This question you look into ma. Photolysis of water molecule yields. Keep it in your mind, where is the exact location of photolysis of water? Photolysis of water takes place in thylakoid lumen. Okay, this occurs in thylakoid lumen. Inside the thylakoid lumen, one water molecule is photolysed to 2 protons plus 2 electrons and an oxygen atom. So, this is what is yielded from one water molecule. Fine. So, it is 2 electrons and 2 protons. Fine ma, it is 2 electrons and 2 protons. Clear, right? Next, this you see, production of NADPH in chloroplast takes place during, okay, production of NADPH in chloroplast takes place during what? Okay, NADPH formation occurs only during non-cyclic photophosphorylation. NADPH formation is not happening in the cyclic photophosphorylation. So, here what will be our answer beta? Production of NADPH in chloroplast takes place during non-cyclic photophosphorylation. Clear, right? This question you look into, which one occurs both during cyclic and non-cyclic modes of photophosphorylation? Involvement of photosystem 1 and photosystem 2 occurs only in non-cyclic, only photosystem 1 is involved in the cyclic mode. So, we cannot opt this option. This you look into, formation of ATP. ATP formation occurs both in cyclic and non-cyclic photophosphorylations. This is true. Release of oxygen. Oxygen is released only in non-cyclic photophosphorylation and it is from photosystem 2. As PS2 is not involved in cyclic photophosphorylation, there will not be the release of oxygen. Formation of NADPH. NADPH is formed at the end of non-cyclic photophosphorylation. Clear? It is not occurring in the cyclic photophosphorylation. Okay, so this is also wrong. Option B will be the correct answer. Which one occurs both during cyclic and non-cyclic modes? It is formation of ATP. Clear, right? Next, this question you see, this is little interesting. It is very simple actually, but looking into the question, most of the students will get confused. Chlorophyll B 650, 660, 670, 678 and all this and chlorophyll A, which is from 680 to 690, represent what photosystem? All these chlorophyll molecules from 650 to 690 are representing what photosystem? Of course, it is photosystem 2. Okay. Photosystem 2 will represent chlorophyll maximum wavelength from 650 to 690 nanometers. If it is photosystem 1, it should be in and around 700 nanometers. Less than 700, it is photosystem 2 only. So, our answer will be option B. Do not get confused when such questions are given. Just choose on that chlorophyll pigment is belonging to the which reaction center. If you can identify the reaction center, then it will be easy to identify the photosystem. Clear, right? Next. Constituents of photosystem 1 are located in. Photosystem 1 is located in PS1 is located in thylakoid membrane when it is undergoing non-cyclic photophosphorylation. PS1 is present in stroma lamellae or stroma membrane. Okay. Stroma lamellae if it is conducting cyclic photophosphorylation. This is in non-cyclic. So, yes, PS1 is present both in cyclic and non-cyclic photophosphorylation. It is present where constituents of PS1 are located on outer surface of grana and stroma thylakoids, that is option C, right. Next question you look into, cyclic photophosphorylation cannot sustain photosynthesis. Okay. Cyclic photophosphorylation cannot sustain photosynthesis because PS1 do not function beyond 680. Wrong. PS1 is functioning beyond 680, that is around 700 nanometers. So, this statement goes wrong. No evolution of oxygen. When there is no evolution of oxygen, that will not affect the electron transport system. Oxygen evolution is involved in other process, but it will not affect the electron transport system. So, this is also wrong. Unidirectional cyclic movement of electron also will not affect the photosynthesis. What is it actually affecting is only ATP is formed and NADPH is not forming. How, ma'am, this is affecting? Because dark reaction or the biosynthetic phase of photosynthesis is dependent on the light reaction. It is dependent on the products of light reaction. What are the products of light reaction? Assimilatory power is the primary product of the light reaction. ATP and NADPH are formed in non-cyclic photophosphorylation, which is helpful for biosynthetic phase, whereas in cyclic photophosphorylation less amount of energy is released, only ATP is released. So, that is how photosynthesis cannot sustain, because less amount of assimilatory power is released at the end of light reaction in cyclic phosphorylation. Clear? So, it will be option D. This you look into, ma? Yeah. Identify the parts marked as A, B, C in the given figure showing ATP synthesis. Okay. A, B, C showing ATP synthesis. Of course, A inside this proton accumulation is happening. So, what is this A side beta? This is thylakoid lumen. Okay. And B, if you see F0 F1 complex or ATP synthase, wherein F0 is embedded inside the membrane and F1 is towards the stroma. F0 F1 complex. Thylakoid lumen, B will be F0 and C will be F1. That is option A. Clear, right? Next question. This you see. Following table summarizes the differences between light reactions and dark reactions. Which of the above pairs differences are incorrect? We have to identify the incorrect pair. Look into the statements properly. Light reactions also called as biosynthetic phase, that is completely wrong. Light reactions also called as what? Photochemical phase. Dark reaction is also called as biosynthetic phase. Light reactions, these reactions occur over the thylakoids. Yes, light reaction occurs over the thylakoids. In dark reaction, these reactions occur in stroma of the chloroplast. Yes, these reactions occur in stroma of the chloroplast. This third statement you look into beta, these produce assimilatory power, that is NADPH and ATP. Clear? This consumes the NADPH and ATP to fix the carbon dioxide. These are directly dependent upon the light. Correct? These are directly dependent upon the light. These depend on the products that are synthesized from light reaction. So, the only incorrect statement is this one. Okay? So, the only incorrect statement is the first statement where light reactions and dark reactions, that is first, option D. Option D will be the correct option. Yes. So, with this we have exercised half of the questions from electron transport system. Tomorrow we will be exercising complete questions of electron transport system and chemiosmotic hypothesis also. So, stay tuned. These questionnaire sessions are really useful for you. Stay tuned, and after completion of all the modules of the questions like statement based, assertion reason, match the following type of questions, diagram based questions, after finishing up this, I will be exercising the NEET previous year questions of this particular chapter. Don't miss that. Okay? So, follow this questionnaire session daily just for half an hour, so that you can listen, memorize and remember it for a long run. Okay? Thank you.